Welcome to the Performance Around the Clock podcast. I am Professor Sachin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to an episode of Performance Around the Clock, and we are recording in Lisbon, Portugal at the Cell Symposium on Exercise Metabolism. And one of the co-organizers of the symposium is my friend Mark Febbraio. He's a professor in Monash University and is a world leader in studying the impact of exercise, mitochondria function as a central regulator of health, and also leveraging this knowledge to treat many chronic diseases from diabetes to cancer. Super excited to be with you, Mark.
Well, thank you, and thanks for the invite, Sachin, to be on the podcast. Hopefully it's a lot of fun.
Yeah, it is a lot of fun. And also this exciting conference that you have organized, I'm enjoying every single bit of it. And I just cannot figure out how to have almost every speaker on this podcast because everybody is presenting something exciting. But the first thing is, I always wonder, most scientists, I ask them, how did they get into what they're doing now?
Okay. So this is an interesting question from my point of view because when I was at high school, I didn't do any science at all. And I was always a very good writer and very good at history and English literature, etc. But my one passion above everything was sport. And I love sport.
Yeah.
So my goal when I was about...
Any specific sport?
All sports.
Oh.
And my goal when I was about 15 was to be a sports reporter, a sports journalist.
Really?
And I actually, in my first degree that I started was in journalism. But I didn't actually have a job, and it was a degree where you had to have what was called a cadetship and then go to school like 2 days a week. And I didn't have a job, and I really thought it was a bit of a waste just to be going part-time to this course. So what I decided to do was enroll in a human movement degree, like a kinesiology degree. And I thought, I'll learn more about sport and biology and physiology, and I'll write as a freelance reporter. So that was my goal. But once I got into my degree, I discovered science. And to the point where I used to walk my teachers with a book to their car, asking them questions. And they would say, well, let me go home, right? And I became very, very interested in physiology and exercise and sports performance, etc. And I also, when I was doing my undergrad degree, started to become fairly dedicated to the sport of triathlon. And I became quite good at triathlon. I mean, I came 51st at the Hawaii Ironman.
Wow.
I was, sponsored athlete, etc. And so after my first degree, I took some time off, and I was just doing triathlon and working in a gym, and and for a few years. And then it was 1989. I'd been racing. I was only 24, but I sort of had this epiphany that I wasn't really going to be good enough to be like the best in the world. And so then I was doing a race in Lake Biwa in Japan in July.
It was hot.
It was hot. It was July 1989, and I was in pretty good shape. And no, 1990. It was 1990, and I was in pretty good shape. I was on this team, this Australian team, and I got off. I didn't have a very particularly good swim, which I never really did, but I was really in shape on the bike, and I rode really, really hard. And I rode into I was probably in the top 10, and I started feeling a bit sick. And the last 30K on the bike was a bit of a struggle, and I got off the bike 13th. So I was still in 13th position going into the run, which was usually my best leg. And I had a diabolical marathon. I ended up on a drip in hospital. Oh. I finished the race, but I was severely dehydrated and heat stressed. So I thought about it, and I thought, well, why did this happen? You know, I need to know more about this. And so I went back to my university, and I went back to my professor, and I said, I'm pretty much done with triathlon. I want to do a PhD. And he wanted me to do this PhD on muscle twitch interpolation or something. I can't really remember. And I said, no. I said, I want to do a PhD on how heat stress affects muscle metabolism.
Wow, that was very personal.
It was very personal, and that was the start of my career.
That's a wonderful, awesome story. Yeah. And from sports journalist to PhD through a heat stroke in triathlon, I had never heard this kind of story. The closest one I have heard, do you know Steve Ostad?
No.
He researches aging.
No.
And his story also, he started as a journalist, but not sports journalist.
Yeah.
He came to PhD, but in between sports journalist and PhD, he was a lion trainer.
Oh, really?
In Hollywood.
Wow.
And actually, he was attacked by a lion.
Oh, really?
And when he was in hospital, that's when I think he changed his mind. So it's kind of interesting.
Yeah. So I continued. I did my PhD, and I continued to do marathon running and ride my bike and everything, but I wasn't really serious about racing triathlon anymore. And then I was working really as a pure exercise scientist. And it's funny how serendipity can change your life. And I had this student, Rebecca Starkey, who Benta mentioned in her talk yesterday. And Rebecca came into my office and she said, I want to do a PhD, and I want to do it in exercise immunology. And I said, I'm not really an immunologist.
Was there a thing like that in the 90s, exercise immunology?
Yeah, so this was in 1997 or 98. And I said, okay, so what do you want to do? And she said, well, I've read a few papers, because she'd done her honours degree with me on heat stress.
Yeah.
And she said, I read a few papers, and I have read that when you exercise, interleukin-6 goes up in the blood. And she said, and everybody assumes that it's coming from immune cells, but no one's really proven that it's coming from immune cells. I said, okay. So I contacted my friend, Mark Hargraves, who I think he was my PhD supervisor, one of them. And I said to Mark, do you know any good immunologists, clinical immunologists? And Mark said, yes, I know that this professor, Jenny Rowlands, at Monash University. So I contacted Jenny. I said, Jenny, we've got this idea. Can we do this? And Jenny said, yep, I'm on board. So Jenny co-supervised Rebecca. And what we actually found in these papers that are, Rebecca's then did medicine.
Yeah.
So she has 5 papers
Yeah.
and she has 3,000 citations, like, you know. So what Rebecca did is she went first and she looked at the spontaneous production of cytokines during exercise and during exercise and heat stress and with beta blockade. And what she found was that the actual spontaneous IL-6 production by the immune cells was not increased but decreased.
Huh.
But exercise primed the immune system to mount a cytokine response. So if you took the blood mononuclear cells out, you cultured them, in the absence of LPS, they made less cytokine. In the presence of LPS, they made more.
Yeah.
So if you're stimulating the immune system with a pathogen, then exercise was anti-inflammatory, which is what we know now. So then we tried.
By the way, just for general audience, LPS is the bacteria.
For the general audience, LPS is bacteria. And so if you have, for example, for the general audience, if you exercise too strenuously in a marathon and you will get contusions to the gut, you may get a little bit of systemic endotoxemia, and that's the LPS, the bacteria coming from the gut. So basically, if you get an infection with a bacteria, your immune system fights it, and this sort of showed that exercise helped in that response.
Yeah.
But we still had to work out where this IL-6 was coming from.
Yeah.
And then we had the same data as Benter, and we started working together, and we discovered IL-6 was the first muscle-releasing factor, what we call myokines.
Yeah.
But the interesting thing, Sachin, is that if Rebecca never came into my office, I don't know, I wouldn't be here today. So
exactly.
I owe her a lot.
No, it's the same thing. Like, for example, when you decided to go to grad school, that was a turning point in your career and also for the field of science. Like,
thank you.
Similarly, there are many students when they decide to do a PhD and they decide to do something, like you said, I don't want to do all that other shit. I want to do heat stroke or heat stress and exercise. Similarly, there are some grad students, they just come and say, no, I don't want to do whatever you are doing. I want to do something else. And those are the ones who open a new field, and if they're hardworking and have the right mentor.
Yeah, and so that happened again in my career, 15 years later, when Martin Whitten, who was just at lunch, came to my lab as a postdoc, and he said, look, we really want to start looking at screening what comes out of muscle, but, it's very hard to do plasma proteomics because, a lot of proteins get sticky. They stick to albumin, which is something that's in the blood. And so he said, why don't we look at what's in, he read papers about exosomes, and he said, why don't we look at what's in exosomes during exercise?
So these are the small
stuff. They're small lipid-bound vesicles that get released from everywhere. And so that started a whole new journey in my career. Like now, that's what we're working on. And it's but going back to the IL-6 story, we then knew that IL-6 did all these good things. It also does bad things. So what we had to do is try and harness the goodness from IL-6 and get rid of the badness from IL-6. And so we worked on this project for 13 years, and we came up with a molecule called IC7. And that molecule turned out to be a therapeutically relevant molecule. And so as my little side hobby is I have a company,
Yeah.
and we are trying to develop new molecules that we hope to get into clinical trials to treat a variety of diseases.
So we know that IL-6, too much of IL-6 is bad. What are the bad things of IL-6 and what are the good things of IL-6?
Okay,
Yeah.
so it's even complicated for scientists, so I'm going to try and simplify it. So IL-6 signals two ways.
Yeah.
Okay, so it has to engage with its receptor called the IL-6 receptor.
Yeah.
Now, the IL-6 receptor is a transmembrane receptor that's in many cells, but not all cells.
Yeah.
Okay, and when the IL-6 engages with the IL-6 receptor, it causes a dimerization, a homodimerization of a signaling molecule called GP130.
Yeah.
And when you get this dimerization and it engages with the IL-6 receptor, and you have a complex now of the IL-6 receptor, and two GP130s, then you get a signaling cascade within the cell, and IL-6 does its thing. And they're usually good things.
Yeah. So it's almost like putting a IL-6 key into the lock.
Correct.
And turning it slightly, then
Correct.
things will start.
Now, I think in terms of evolution, the body has learned that in certain situations, you don't want to activate this signaling cascade. So most pro-inflammatory cells, particularly immune cells, they don't express the IL-6 receptor. But what happens in disease, we have these molecules which are called metalloproteinases. Now, for the layperson, just think of them as a lawnmower.
Yeah.
And they come along and they cleave off these receptors. And so the IL-6 receptor becomes soluble.
I see.
And the soluble receptor then binds to the IL-6 in the blood.
Yeah.
And then that can signal in cells that don't express the IL-6 receptor. And that's called IL-6 trans-signaling. And that's the bad thing about IL-6.
I see.
And there is my dear friend and colleague, Stefan Rosejohn, he figured all this out. And he developed a drug called soluble GP130.
I see.
And that then, if you inject it, then that will bind the IL-6 and the IL-6 receptor in the blood and neutralize it.
I see.
And that drug is marketed as a Lancacept, and it's in a phase 3 clinical trial for ulcerative colitis. And it's working. And it looks like it'll work for many diseases, including atherosclerosis, etc. So on one hand, you want to block IL-6 trans signaling, but on the other hand, you want to maintain membrane-bound signaling.
I see.
And that's exactly what we did with IC7. So what we did with IC7 is we took the part of the molecule that binds one of the GP130s, but not the other.
Yeah.
We took it off and we replaced it with a sequence from another cytokine from the same family called CNTF. And that motif does not bind the IL-6 receptor, it binds the LIF receptor.
I see.
So now, if you give, and that's why it's called IC7, interleukin 6, CNTF, and the first 6 didn't work.
I see.
So it was the number 7, right? So we call it IC7. It'll probably be called something else, right? But that's what we called it. And so if you give IC7 to we haven't done it in humans, but, we've done it in monkeys and we've done it in mice. And if you give that, it's anti-inflammatory because it can't trans-signal.
Oh, I see.
So we're harnessing the goodness of IL-6 and blocking the badness of IL-6.
Wow.
So that's something that I've been working on for 15 years.
Yeah. So that's does that explain this IL-6 mechanism? Explain why exercise, at least partly explain why exercise helps to lessen the burden of many inflammatory diseases.
Correct.
Yeah.
And if you exercise, as Benta Peterson showed yesterday, if you use a pan IL-6 inhibitor, you can blunt many of the positive effects of exercise.
Yeah.
And if you use an IL-6 knockout mouse and you exercise it, it doesn't have all the benefits as you.
Yeah.
Right.
Yeah.
So one experiment that we did back in the early 2000s, and we probably couldn't do this experiment again. It was Benta's idea, and it was and it was Rebecca's study.
Mm-hmm.
So what we did is we wanted to see if exercise was truly anti-inflammatory.
Mm-hmm.
So Benta is a professor and works in infectious diseases.
Yeah.
So we were able to get through ethics this experiment, and we were able to inject healthy humans with a very, very small amount of endotoxin,
Yeah.
of LPS.
Yes.
And I was a subject in this. You didn't even know.
Yeah. Yeah. Yeah.
But what it did, it increased the TNF response.
That's it.
So what we did is we did a randomized control study. So we did either no exercise or exercise with the LPS,
Yeah.
and we measured the increase in TNF. And when you exercised, there was no increase.
Oh, so TNF is the bad cytokine.
It's a bad cytokine.
Yeah.
And it was elevated in every subject if you did not exercise.
Yeah.
So then we said, okay, is it the IL-6 that's doing this? So what we did is we measured the IL-6 response during exercise, and then we did a third trial. And in that third trial, we just had the same people resting, gave them the endotoxin, but this time we infused IL-6 to mimic the levels we saw in exercise.
I see.
No TNF.
Okay.
So that was published.
Yeah.
And, that was a paper we sent to Nature and, and
This is a human study too.
it was a human story, and no one really published human stories back then. They weren't really important, right? They weren't in vogue. But, we said this is the first irrefutable evidence that exercise is anti-inflammatory.
Yeah.
Anyway, it ended up in the FASEB journal, and it's had thousands of citations.
Yeah.
So, and it's Rebecca's, as I said, she has 3,000 citations, about 1,000 of this paper.
No, this is really exciting because, during COVID, one thing we all remember, of course, the people who had comorbidity, diabetes, hypertension, they were more prone to severe side effect. And then even young people. There are many athletes who were playing, who were practicing in isolation, but there is I don't recall any athlete getting so seriously sick with COVID to get hospitalized.
No, I think, it's a bit hard now to, what we need to do is really look at who got sick before vaccines.
Yeah.
And definitely those people who were elderly, obese, had comorbidities, they're the people that were hospitalized and unfortunately died.
Yeah.
Younger, healthy people, they didn't really. And, it caused it wasn't a very good period of anyone's lives.
Yeah.
But it wasn't it wasn't a very good period in my life because I think COVID brought out the best and the worst in people.
Everybody.
Right? So the best were the people who were on the front line, nurses, your heart bled for these people.
Yeah.
But then you had younger people going, well, you're taking away my civil liberties, blah, blah, blah. I'm not going to get sick. And, and I would say to some of these people, but what about if you got it and you gave it to your grandma?
Yeah.
do you love your grandma?
Yeah.
Well, think about other people's grandma. So, COVID was it. But definitely exercise seemed to provide some immunity.
The way I was pointing out was, there are many sports teams in the NFL, NBA in the US. They were practicing within this bubble.
Yeah.
And once in a while, some player might get positive.
Yeah.
But then no one was actually serious. Many of them were not symptomatic. And I was always wondering, how come these athletes?
Yeah. So exercise and immunity is a really interesting phenomenon because, if you think about, people who are truly, the founders of this field, people like David Neiman and in North Carolina and Benter and Laurie Goetz and Jeff Woods and all these immunologists, they will tell you that it's a J-shaped curve.
Yeah.
So if you exercise, if you think about the J-shaped curve, that if you exercise moderately and regularly, then your protection is increased.
Yeah.
So your relative risk goes down. Yeah. But then if you overreach, then you can smash your immune system. Yeah. And it's interesting because when I was an athlete, I didn't get many injuries. Yeah. I was a very, very hard trainer. I didn't consider myself to be that talented an athlete, so I had to work really hard, and I got sick a lot. Wow. A lot. Like I never really got injured, but I was always getting upper respiratory tract infections, and I think my immune system was pretty smashed. Yeah. Because I was just overreaching all the time.
So then the question is, does anyone know why over-exercise reduces or suppresses immune system?
I think it's got to do with the fact that if you this is well documented, that athletes, I would say, based on the clinical guidelines, probably 40% of highly trained endurance athletes have neutrocytopenia.
Really?
Yes. And I have...
Can you explain neutrocytopenia for everybody?
So that means neutrophils are one particular white blood cell that is known to be there to fight infections. Yeah. I think it's to do with the turnover of the cells. Yeah. Like even now, I'm a habitual exerciser, and my neutrophils are right on the borderline of being mildly neutrocytopenic. Like because I have a family history of heart disease, and I have some coronary calcium in my right coronary artery. So I see a cardiologist every 3 months or every 6 months, and I get my bloods done. Yeah. And and my bloods have always been really good, except my neutrophils are always a little low.
A little low.
Yeah. Sometimes they go up, sometimes they go down a bit. They never go plummet. But so I think one of the mechanisms by which athletes can be susceptible to infection is because they have low neutrophils.
Oh, I see. So, but that connection, why or how? Well, too much exercise reduces.
Well, I think it's. I don't know. Yeah, I'm not an immunologist, so I don't know. But I think it's got to do with the impaired turnover. So cells, live and die, and I think that they just coming from the stem cells coming from the marrow. Just I don't know the exact reason, but certainly it's well documented. Yeah. And my cardiologist, I knew him through triathlon. Yeah. And he told me this as well. So
no, that's something that
because when he looked at my neutrophils, I said, "Should I be worried about that?" He goes, "Nah, 40% of people who exercise have this." So anyway.
So what is exciting now for you?
Sachin, the most exciting thing for me is training my mentees. I mean, it is really gratifying. I think as you become older
Yeah.
and a little bit more successful, you start to breathe a little easier about, where you are in the lexicon of science.
Yes.
And then you start to think about the next generation.
Yeah.
And you start to think about, making the next generation excited about science,
Yeah.
positive about science. And that's exciting. In terms of my own research, I think the work we're doing on extracellular vesicles is super exciting. And the stuff I'm going to present this afternoon, I'm so excited about. It's basically a story where we've figured out how exercise can slow the progression of metastatic breast cancer. And
Wow.
I'm super excited about this.
Yeah.
We're about to submit the paper. I'm really excited about this work. And now what we're trying to do, in collaboration with Jonathan Long from Stanford, we're going to map the whole exercise EV landscape by using these turbo ID mice, where it's not really important, but it's just we can work out where the cellular origin of a protein within an EV is. We can work out, did that come from muscle? Did that come from liver? Did that come from the gut?
Yeah.
So we're going to figure it all out. And that'll take me to retirement.
No, that's a exciting time because the idea that exercise produces cytokines or exokines, now the idea is actually there may be hundreds or even thousands of different things.
So we have a paper in review at the moment.
Yeah.
One of the things that we're all of us are, committed to exercise because we see the benefits in our work. But we've got a paper in review at the moment that is pretty exciting. We exercise trained a mouse model of Alzheimer's disease. And we found that the exercise training improved cognition in the mice.
Yeah.
And just the way that we did it, we used this thing called a Barnes maze.
I see.
Now, let me just explain what a Barnes maze is.
Yeah.
So a Barnes maze looks like a roulette wheel.
Yeah.
Except where you've got the numbers, there's holes.
Yeah.
And there are holes, maybe on a roulette wheel. Let's say there's 80 of these, and there's like 5 holes.
Yeah.
And only one hole is an escape route.
Yeah.
And that's the hole the mouse wants to find.
Yeah.
So what we do is we put the Barnes maze in a room like this, and there are visual cues and so there might be a red cross.
Yeah.
Now, if the hole, and then you bring the mouse in, and over 4 days, it goes in there every day, and it learns where the exit hole is, and it goes, it's next to the red X.
Yeah.
So then you take the mice away for a week, and then you bring them back. And if it's got good long-term memory, it'll have less errors to holes because and then it'll figure out it's near the X, and it'll go down. Yeah. So when you exercise train by giving running wheels to these Alzheimer's mice, and you do that experiment, the error to holes are less if you exercise.
Wow.
So then what we asked was, are the EVs responsible for this?
Yeah. And
we did an experiment where we isolated EVs from exercising mice or sedentary mice, and we then we gave them to these recipient mice with Alzheimer's disease, and not all of the effects were due to the EVs, but partially the EVs could partially explain the benefit of the exercise.
Wow, that's exciting. So you're taking the plasma from
we're taking the plasma from an exercising mice, purify. So we find this in a number of scenarios. So what when you said what excites me, my holy grail, and we're starting. We've got this evidence in breast cancer. We have some work that we're doing together with a collaborator at NYU, looking at pancreatic cancer. So my idea is, if somebody gets diagnosed with a cancer in the early stages, when they're healthy, they can exercise. You can purify their EVs.
I see.
Store them, and then if they're undergoing, chemo or immunotherapy where they're a little bit fatigued and can't really exercise, then you can inject their own EVs into them.
That's exciting.
That's my idea.
Yeah, that's very cool.
I don't know if it's going to work, but that's my idea.
No, it always starts with some hypothesis and hope.
Yeah.
So, thank you, Mark.
Thank you.
Very exciting.
It's been a pleasure.
Yeah.
Thank you.
Thank you.